I'm Eric Nestler. I'm at Mount Sinai School of Medicine in New York City, and it's my great honor to be here today to interview Dr. Paul Greengard. We're sitting in Paul's office at the Rockefeller University. I had the privilege of being a graduate student in Paul's lab in the late 1970s and early 1980s, so it's particularly fun to be here today. Paul, thank you for joining us.
Thank you, Eric.
I thought we'd start by talking about the contributions that your laboratory has made to the field of neuropharmacology, and start with the early days when your lab started looking at the role of cyclic nucleotides in the brain. What prompted you to take that focus?
What prompted that was the discovery by Earl Sutherland and his colleagues that the hormones, norepinephrine and glucagon, raise the level of cyclic GMP in liver and muscle cells. Mm-hmm. And the thought occurred to me that maybe nerve cells communicate with each other in a manner similar to that by which the hormones communicated from a sending cell to a receiving cell. Despite the difference that the hormones are working over a matter of, in some cases, feet, and these things are through millionths of a centimeter. And I remember having that thought many years before I did anything about it. I was doing my postdoctoral studies, and then I was in industry for 9 years, pharmaceutical industry. So when the first thing I did when I went to Yale was to see whether neurotransmitters might raise cyclic GMP, and we found that there's several: dopamine-sensitive adenylate cyclase, octopamine-sensitive adenylate cyclase, serotonin-sensitive adenylate cyclase, and it was clear then that the principle that the endocrine system had evolved was also true for synaptic communication across a tiny synapse.
And then you also took analogies from the earlier studies in endocrinology to pursue how it was that cyclic nucleotides then produce their functional effects in nerve cells with the discovery of protein kinases in the brain.
Right. That became clear many years after the Sutherland discoveries, when, as a paper by Wallace, Perkins, and Krebs showed that the cyclic GMP works through an enzyme which at the time was called phosphorylase kinase, and the name was then changed to cyclic GMP-dependent kinase. And I wondered whether the same thing might happen in the brain, that there was a kinase involved in mediating the actions of neurotransmitters. And so we looked for such an enzyme in the brain, and we found that it was present in much higher concentrations than anywhere else. And in addition, it was associated with the plasma membrane, so everything was in the right positions to make that a viable hypothesis that protein kinase mediated the actions of neurotransmitters. And the argument about whether cyclic GMP was important was finally resolved. We published 2 papers back-to-back in PNAS, one with Felix Strumwasser at Caltech, and the other one with Eric Kandel at Columbia, in which we showed that injecting the catalytic subunit of cyclic GMP-dependent protein kinase into nerve cells mimicked the effect of the neurotransmitters. So that was pretty definitive evidence.
I was in your lab at the time that those studies were published, and I remember being struck by the reaction of the field to your lab's proposal that cyclic GMP and cyclic GMP-dependent protein kinase are important in regulating neural phenomena. There was a lot of skepticism. Would you like to talk about that?
The peak of the skepticism came when I gave a lecture in front of a very large audience, and after I finished my talk, somebody got up and said, "This is absolutely heretical thinking and should not be allowed." I thought that was a little bit excessive, but that was the bottom. But then gradually people began to accept it. I think.
Was there a turning point, you think, that, or was it just more of a gradual process?
I think it was a gradual thing. There was good news and bad news about people not accepting this. The good news was that I didn't have any competition for 15 years, from the late 60s to the early 80s. The bad news was I was sort of unpopular conceptually in the field. But the good news outweighed the bad news, so we're able to develop this story incrementally, but over that 15 years, the evidence became overwhelming, and it was hard any longer to deny it. Ironically, the first place we showed the most compelling evidence that neurotransmitters could work through these pathways, we were looking at nucleated erythrocytes, bird erythrocytes, and we're able to show that when the neurotransmitter, I believe it was norepinephrine, I'm not sure, activated its target cell, there was an increase in Cyclic GMP in those cells, and the Cyclic GMP could produce the effect on the ion channel, could mimic the neurotransmitter in producing that. That was pretty compelling, and then we went on to show the same thing in nerve cells. It was around a gradual build-up, and then we were looking for substrates for this pathway, and found a few of those, and found other kinases, so increasingly the evidence became stronger and stronger.
I'd like to ask you about those other kinases, because I remember that being part of the process by which your lab established an important role for protein phosphorylation. After the finding of cyclic GMP dependent protein kinase in the brain at high levels, I believe your lab went on to discover cyclic GMP dependent protein kinase?
That's correct. The way we found that, I wanted to purify the cyclic GMP dependent protein kinase. So we put this extract over a column, and we got our two peaks. And at that point, people had gotten very interested in other cyclic nucleotides, cyclic AMP, cyclic GMP, cyclic, all the nucleotides. And so we had this second peak, and so we compared the two peaks and found that cyclic AMP activated the kinase activity of the first peak, and cyclic GMP activated the activity of the second peak. And then we, by dissecting that system, realized it was a totally different protein kinase regulated by cyclic GMP and not cyclic AMP. Now that too became a controversial issue. It's interesting in retrospect. Some of the people down at Vanderbilt who have been fellow students, if you will, of Earl Sutherland, really didn't like this. They said this is some denatured form of cyclic AMP dependent kinase, which now saw cyclic GMP dependent kinase as the activator, not cyclic AMP as the activator. So it was interesting.
Absolutely. And then shortly thereafter, your lab discovered what has come to be called calcium calmodulin dependent protein kinase 2, among other calcium kinases. And one, yeah.
It was very similar.
And 3, I believe, perhaps, right? Yeah.
No, we didn't have 3.
Okay.
That came later. But it was the same thing. We knew there was calcium-dependent protein phosphorylation. So we wanted to see how that was happening. And what we did is we ran an extract over a column and found there were 2 peaks of calcium-dependent kinase activity present. And the one we call calcium-dependent protein, what we changed the name later to calcium calmodulin-dependent protein kinase 1 and calcium calmodulin-dependent protein kinase 2. And today we know that CAM kinase 2, as it's abbreviated, is much more important. It's an important component of the postsynaptic density and plays a vital role in synaptic transmission.
I think it's hard for students who grow up in biomedical science today, who by second nature know that protein phosphorylation mediates so many different neural phenomena, to think back, not that long ago really, 30 or so years ago. Remember, you published a paper in a review in Science in '78, where I believe one of the first times you proposed a diversity of kinases with mediating a diversity of types of signals on many types of neural phenomena. And now the human genome sequencing has indicated there's about 500 or so protein kinases. So I don't know if you want to comment on that perspective.
Well, there's now over 1,000 tyrosine protein kinases, which is one subclass. I think just a lot of evidence accumulated, not only in our lab but in many other laboratories. And now with the modern molecular biology, we can knock out genes or mutate them and so on. And you know, the evidence has become incontrovertible. And now, as you say, it is second nature to people.
I mean, it really has transformed, I think, the way young people approach a biological question, because protein phosphorylation is almost a reflex in terms of a mechanism that's pursued.
Yeah. Now that's what's come a long way from the... See, at the time we started this work, it was accepted that chemicals, the way nerve cells communicated with each other was through neurotransmitter release, diffusion to this postsynaptic membrane to activate receptors, which at the time were hypothetical when we started this. And that was a battle that went on for 40 years, and it was won pretty resoundingly by the chemical school. A very distinguished scientists such as Eccles, who took a long time to accept it. He believed in the electrical transmission, which is perfectly logical. The idea was that when the nerve impulse came down to the ending, there was an electrical field generated by the nerve impulse, which changed the membrane potential across the postsynaptic membrane and initiated or inhibited a postsynaptic response. But as I said, the debate was won rather resoundingly by the chemical school. It was about this time we started our work, and at that time people believed, okay, it's a chemical, but it's an electrical response to an opening of a voltage-gated ion channel, which initiated the signal going down the postsynaptic cell. And there was reason to believe this. The things that did that were, for example, acetylcholine receptors opened up ion channels. Those studies were perfectly compatible, what was known at the time, with the idea that all that was involved was a change in ion conductivity across the membrane, and therefore an inhibitory or an excitatory postsynaptic potential. But again, as we discussed earlier, I thought it might be more complex and it might be somewhat like the hormones, and that turned out to be the case. You can say both ideas were correct. You can think of that electrical signal as being how the fast excitatory neurotransmitter glutamate and the fast inhibitory neurotransmitter GABA produce their effects. But then there are over 100, probably many, many more neurotransmitter pathways which work through these very complicated signal transduction cascades involving changing the level of the second messenger, activation of a protein kinase or a protein phosphatase, changing the phosphorylation of a key substrate protein, and the production of a physiological response.
You alluded to earlier, Paul, that the next step after identifying protein kinases was to find the substrate proteins through which the protein kinases work to produce their functional effects.
Right.
Let's talk a bit about the first substrates that your lab discovered in the brain, which was a protein that I was involved in. We called it Protein 1 at the time. Maybe you can tell us about Protein 1.
Sure. So after we'd established that the neurotransmitters work through changing the level of a second messenger, and the second messengers work through activating protein kinases or protein phosphatases, the next question came, what are the substrates for the kinases and phosphatases? And in those days they had just developed these very primitive techniques for running gels and doing Western blotting and doing determination of radioactivity on the gel. And what we did was we looked for, we used radioactive ATP and looked for substrates that might be radioactive. And then we found this protein which we called synapsin 1, with two of them, synapsin 1 and protein 1 and protein 2. And it turned out, as we learned more about protein 1 and purified it, that protein 1 was associated with a nerve terminal. And as we subsequently learned, it coats all synaptic vesicles and regulates neurotransmitter release in a very profound and dramatic way. So that was synapsin 1 and synapsin 2. And the reason, in retrospect, that we found them first was they're basically present on all synaptic vesicles in all nerve terminals in the brain. And so they're enormously abundant. And so with the primitive techniques that were available at that time, they were the first ones we saw because they were the most abundant.
Right. And in fact, the discovery of synapsin, I think, and demonstrating its concentration in nerve terminals and its role in neurotransmitter release was very important in stimulating the field to elaborate a biochemical process of neurotransmitter release, which had, before that discovery, remained completely opaque.
Right. So yeah, that's true. Once we found out that this protein 1 was synapsin 1, which got phosphorylated, then we compared what synapsin 1 in the phosphorylated form did compared to the dephosphorylated form. And what we found was that the dephosphorylated form of synapsin 1 inhibited the vesicles from fusing to the plasma membrane and releasing their neurotransmitter. The phosphorylated form did not do that. So by phosphorylating, you remove this inhibitory effect and allowed an increase in neurotransmitter release.
Mm-hmm.
And it turned out that there are these two classes of synapsins, which even to this date is not quite understood why they're, because there's synapsin 1A, synapsin 1B, 2A, and 2B. It's not quite clear why one needs so many of these synapsins to do what the nerve cell does, because by and large the ratios are pretty much the same amongst all different nerve terminals.
Mm-hmm.
Ivar Wallace and Angus Nairn, two postdoctoral fellows in the laboratory, took on the following project. We knew that synapsin 1 was present in every nerve cell in the brain. We asked whether there might be certain phosphorylated proteins, which are present in certain regions of the brain and not in others, because the brain is so heterogeneous.
Mm-hmm.
I have to say that also in retrospect, it's this discovery of these phosphoproteins and these signaling pathways that provide overwhelming proof for the heterogeneity of different nerve cells. Like when I started out, it was like a nerve cell is a nerve cell is a nerve cell, despite the fact that Ramon y Cajal had shown these hundreds of different types. Anatomically, what our work did was to show there are hundreds of different types biochemically. So it did help in that regard.
Another substrate that your lab discovered a number of years ago that remained a major focus for a while was DARP32. Would you describe for us what led your lab to discover that protein?
So we were looking for region-specific phosphoproteins. And we found that when we took the neostritum, we found a whole bunch of these different phosphoprotein bands, which you didn't see, for example, in the cortex. The explanation for that, retrospectively, was that the striatum is very homogeneous compared to the cortex. Cortex has a lot of different cell types. The striatum has basically these two major classes of what are called medium spiny projection neurons. In the cortex, there are all these cell types, and therefore all these different phosphoproteins which didn't stand out. In the striatum, there's two cell types basically that stand out, and these two cell types, which are very similar in many respects, have a large amount of this one phosphoprotein type, and we're able to see it because of the homogeneity. It wasn't diluted by lots of other signaling types. And the DARP32 turned out to be very important in dopamine signaling.
Hence its name.
Yeah, the DARP32 is an acronym for dopamine psych-AMP dependent, or dopamine psych-AMP regulated phosphoprotein of 32,000 molecular weight.
And more recently, your lab had shown that the protein's role is far broader than simply mediating the effects of dopamine.
Yes. The reason we focused on the dopamine initially was when we found this molecule selectively in the striatum, we thought maybe it was found by looking for psych-AMP dependent protein kinase substrates. Because dopamine was such a prominent neurotransmitter there, we then showed that dopamine regulated it. And given the clinical importance of dopamine in many psychiatric diseases, Parkinson's, schizophrenia, ADHD, we studied the dopamine system a lot. The other reason for focusing on the dopamine system for a decade or more, actually 2 decades, was that the concentration of DARP32 in these principal cell types in the striatum was enormously higher than in the rest of the brain. So if you did immunostaining, immunoperoxidase staining, the striatum was black and the rest of the brain was hardly labeled. It turned out that this tiny amount of DART32 in the cortex, for example, was as vital for those cells in the cortex as it is for the striatum. And the reason why it's, even though it's present in tiny, tiny concentrations, the reason it's effective in the rest of the brain is because DART32 is an incredibly powerful molecule. In its phosphorylated form, it inhibits protein phosphatase 1, the major serine threonine phosphatase in the brain, at 10 to the minus 10th molar, which means a trace amount of DART32 can fully inhibit this vital enzyme, the protein phosphatase.
And thereby control the state of phosphorylation of hundreds of other proteins.
Right, right.
One of the interesting historical findings, I think, was the observation that several of these substrates, like synapsin, like DART32, are phosphorylated by several kinases at either the same or different sites. And at the time, I think it revealed a completely new level of molecular convergence of regulation. And I want to talk about the implications of that, perhaps.
Yeah. Okay, so it's true. We found, each one had its own story, but we found several phosphorylation sites for different kinases. There were 4 major ones, and now we know 3 minor ones, which we've barely begun to mine. But the 4 major ones...
This is on DART32.
On DART32. And we found that these 4 major phosphorylation sites on DART32, they were phosphorylated by different protein kinases in response to different neurotransmitters. So it became clear whether this is a way of these different neurotransmitter pathways interacting in the DART32 containing cells. And one of the ways they interact is very simple. When one of these, the Cyclic site, the dopamine Cyclic site, was a residue called threonine 34. And when that is phosphorylated, you got this very potent inhibitor, protein phosphatase 1. The other 3 sites were phosphorylated. The consequence of phosphorylation on those other sites was to affect the efficiency of either phosphorylation or dephosphorylation of this threonine 34. So that the other neurotransmitters are now modulating the dopamine signaling pathway through this complicated intramolecular interaction in the DART32 molecule.
Yeah, leading to very complicated modes of cellular regulation.
Right.
It's just a fascinating story how these biochemical networks of information control have evolved.
Yes. So it turns out that this is a major mechanism, if not the major mechanism, by which these different neurotransmitter pathways interact with each other. So you've got this convergence of all this signaling onto a single molecule, the DART32, through the neurotransmitter receptors, second messengers, kinases, onto DART32. And the DART32, in turn, by regulating the activity of protein phosphatase 1, controls the state of phosphorylation of virtually every downstream protein, all the effector proteins. Remarkably, there's hardly a protein in the brain that exists that is not regulated by protein phosphatase 1. So you have this convergence onto the DART32, and then a divergence going out from that. Interesting, we found another molecule recently which plays the same role for calcium that the DART32 plays for Cyclic GMP. And that's a molecule we call regulator of calmodulin signaling. And that also integrates a whole bunch of stuff and interacts with a bunch of stuff that goes through the Cyclic GMP pathway.
People might know that as RCS protein.
Right. Regulator of calmodulin signaling, RCS. It was also, we found it in the same original studies that we talked about earlier. When we were looking for substrates for Cyclic GMP dependent kinase that might be region specific, we found these several bands on a gel. I mentioned that earlier. Had several molecular, about 6 of them. Were very interesting. We were so consumed with the DART32 for 15 years that we didn't go to these. And then we turned to those others and we found another one of these. It was ARP21 or DARP21. And that was regulated by the calcium pathway. And interesting, this RCS, or ARP21, controls the state of dephosphorylation, one of the residues on DART32. So it's a regulator of this master regulator molecule. It's a way of, it was one of the mechanisms by which the calcium signaling pathway controls the Cyclic GMP signaling pathway.
Paul, let's turn to the focus of your lab in more recent years, when a major interest of the group has been Alzheimer's disease.
Okay.
Maybe you can describe a little bit about what drew you to focus on Alzheimer's disease, and in particular talk about your recent discovery of a major protein that controls amyloid deposition in the brain.
Right. So I mentioned a few minutes ago, because of this vastly increased amount of information we had about signal transduction pathways, I thought it might be possible to understand the molecular basis for various neurological and psychiatric disorders, such as depression, schizophrenia, Parkinson's, and Alzheimer's.
Mm-hmm.
And to try to gain insight into the mechanism by which the drugs that are used to treat those disorders achieve their effects. With regard to your specific question about Alzheimer's, it was known that beta amyloid was made in the brain by converting a molecule called the amyloid precursor protein into an intermediate amyloid precursor protein C-terminal fragment, and that was then converted to beta amyloid. These are two steps requiring two enzymes known as beta secretase and gamma secretase. So we were able, taking cell line N2A cells, to permeabilize them and show that we could still make beta amyloid in these broken cell preparations. But that was exciting because we could now start controlling and characterizing the enzymes. So we now had these broken cell preparations. We found that they could make the beta amyloid in this two-step cascade. We were then able to dialyze all the interior of the cell out and show you loss of the ability to make beta amyloid. If you added back ATP, adenosine triphosphate, you're able to accelerate the conversion of the APP, the amyloid precursor protein, to beta amyloid. We then showed that was due to the second step, the gamma secretase. And then we kind of left that on the shelf for several years, and then a postdoctoral fellow in lab, Bill Nestler, picked up the gauntlet on this and wanted to see what the ATP is working on. And so the way this project was approached was to take drugs which are known to work by competing with ATP at binding sites. This is mainly these tyrosine kinase inhibitors, which are used very extensively now in developing anti-cancer drugs. And one of them, Gleevec or imatinib, which is made by Novartis and is now used to treat people with chronic myelogenous leukemia. This Gleevec was able to prevent the ability of ATP, adenosine triphosphate, to catalyze the conversion of the APP, the amyloid precursor protein, to beta amyloid. We published that. I was a bit concerned about it, but these younger people have to get their papers out. I was concerned about it because I thought once we showed this, that this Gleevec could block the formation of beta amyloid, that the whole pharmaceutical industry would all come in, and they didn't. Part of the reason for that was one pharmaceutical company published a couple of papers saying this was an artifact, which we knew what it wasn't. But it bought us time. It kept other people from muddying the subject. And so we went on with this, and we were able to show that the Gleevec bound to a protein which we today call gamma secretase activating protein, or abbreviated GSAP, an acronym. What happens is that, as it turned out, that this GSAP, or gamma secretase activating protein, binds to the C-terminal fragment of the APP molecule. It then forms a ternary complex with the gamma secretase. So you have the enzyme, the substrate, and this modulator, the GSAP, which activates the cleavage to form beta amyloid. There's one of the problems in developing inhibitors of gamma secretase to block beta amyloid formation is that they cleave some other pathways, most notably Notch. And so most of these gamma secretase inhibitors have not only, by inhibiting gamma secretase, not only prevented the formation of the bad substance, beta amyloid, but they've also prevented the formation of vital substances. Fortunately, with a GSAP, it only activates only the beta amyloid pathway, not the Notch pathway. And that was great because it means that inhibitors of GSAP can block beta amyloid formation without blocking notch formation. And if you put that in a broader perspective, what it's telling us is that the GSAP is acting like let me back up. There are many regulatory proteins known now which regulate proteases. Proteases are very promiscuous, but they require a regulatory protein to, say, attack this substrate and not this one. And that's what the case is with GSAP. It's exactly a classical regulator of protease. The GSAP regulates the ability of the gamma secretase to cleave this APP-CTF to form beta amyloid. It does not affect the notch pathway. So now there's a whole new area.
It's an exciting new area for drug discovery.
For where you can look at inhibitors of GSAP. And we have made compounds which are very potent selective inhibitors of beta amyloid and not affecting the notch pathway. And we've made knockout mice of GSAP, and they no longer form plaques. They no longer form these beta. The amount of beta amyloid is greatly reduced. There are no plaques formed in the brain, but there's no toxicity either, as you can look at. So it's a kind of intestinal pathology they looked at when you inhibit the
to look for notch-related,
of the notch, NICD it's called. So this is a nice selective thing, and we're now making compounds which are much more potent inhibitors of the gamma secretase pathway selectively for the beta amyloid. So I think this is going to be a very potent way to get into exciting new drug development. We have compounds which are more potent on the in terms of interacting with GSAP. Oh, I neglected to mention that in this ternary complex of the GSAP, the gamma secretase, and the APP, what happens is that the ATP is required. It causes a conformational change in the GSAP, which enables it to activate the cleavage. And that the Gleevec competes with the ATP binding site on the GSAP. And we've just now identified the residue that the ATP binds to, so we're very excited
which will
about
help
that.
drug discovery even further.
Even more.
One last discovery I'd like you to talk about before we discuss other subjects is your lab's focus on a protein called P11, and which you've implicated in depression, leading to potential new therapies for that syndrome as well. Could you tell us how your lab came across P11?
Yes. It's pretty well established that virtually all, well, certainly the SSRIs, the selective serotonin reuptake inhibitors, but even some of the other antidepressants like the monoamine oxidase inhibitors and the tricyclic antidepressants appear to have their antidepressant effect through increasing signaling at serotonin synapses. And they do this by increasing the amount of serotonin in the synapse and not affecting the postsynaptic serotonin receptor. We wondered if, and in this case this is the work of Per Svenningsson and Mark Flagellé, we wondered whether there might be endogenous proteins in the plasma membrane of the serotonoceptive cells which might modulate the serotonin receptor. So using a yeast-2 hybrid technology, which Mark Flagellé had introduced into our lab, he had done his graduate studies at the Pasteur Institute in a laboratory that was one of the major developments of yeast-2 hybrid technology. So when he came here, he started working with Per on this project. Can we, using the serotonin receptor, find a binding protein which might regulate its activity? And he found this protein, P11. In contrast to the GSAP, which was totally unknown, it was not even in the human genome at the time we found it, the P11 was a well-known protein, but there was no evidence that it was involved in brain function. So here we found this P11 to be a binding protein in the plasma membrane for certain serotonin receptors, not all. And so then the question was, is this a possible biological relevance? And we found that it co-localized with the serotonin receptors, or certain of them. And then we found, most excitingly, that the P11 was a chaperone which it caused accumulation of the serotonin receptor in the plasma membrane. We still don't know whether it's due to an increased recruitment to the membrane or decreased endocytosis, but that's one of the things we're working on. Now, this was terribly exciting because all of the previously known antidepressants work by increasing serotonergic transmission, by increasing the level of the neurotransmitter serotonin, with no effect on the receptor. Here we had a protein which did the opposite. It increased the amount of the serotonin receptor in the presence of a fixed amount of the neurotransmitter serotonin. So this seemed very exciting, and we wondered whether, in fact, there might be a relationship. We showed in a number of ways, for example, changing the depressive state or an antidepressant state of animals caused a change in the P11 levels. For example, in animal models of depression, the level of P11 was way down. Post-mortem human brain tissue, depressed people with major depressive disorder had a much lower level of P11 than normal controls. Conversely, we could manipulate the state of P11 and get these really dramatic behavioral effects. For example, if we knocked out the P11, the animals had a major depression as judged by all of these behavioral studies. Now, a lot of people who are not familiar with the field say, how can you judge from a mouse's behavior or a rat's behavior whether this is a good predictor of depression in a human? But the fact is, there's a good correlation, as you know well, Eric, from your own work, there's a good correlation between the ability of different drugs to be clinically effective and their ability to do these, to have these effects on behavior in rodents. So we knocked out P11 constitutively and got this depressive behavior. If we overexpressed P11, the animals behaved as though they'd been given an antidepressant. We then, in a series of studies with Michael Caplet, who had been an MD PhD student here and is now a brain surgeon across the street at Cornell, we did studies to localize the region of the brain in which this might occur. And we found if you did not the constitutive knockout, but only knocked out the P11 in the nucleus accumbens, a known system for reward and mood regulation. If you knocked that only in that region, you got a depressed animal. We then went on to show that within that region, this is the work of Jennifer Warner-Schmidt, if you knocked out the P11 only in one cell type in this relatively small region of the brain, the nucleus accumbens, you mimicked the constitutive knockout. We were able to identify a single nerve cell type where this happened.
And in fact, it's a minority of cells in this entire region that is mediating this effect.
1% of the cells in a small region of the brain, you remove P11, you have a severe depressive phenotype. We were very excited about this. We were able to take constitutively knocked out mice, so there's no P11 anywhere in the brain or body, and put the P11 back into this one cell type, these interneurons that represent 1% of the cells in the nucleus accumbens, and totally restore normal activity. So this has been a rather rewarding series of experiments. We now know of P11 enriched cells in several parts of the brain. Interestingly, they're in studies in collaboration with Eric Schmidt, a postdoctoral fellow in Nat Heinz's laboratory, and Jennifer Warner-Schmidt in our laboratory. We just found in a paper that just came out in Cell last week that there's a cell type in the cortex, in layer 5A in the cortex, which is highly enriched in P11. If you knock out the P11 there, you get the opposite of what you get if you knocked out in the nucleus accumbens. And if you knock it out in the cortex in these layer 5A cells, the animals show no depressive phenotype, but they no longer respond to an antidepressant substance with a behavioral change. This is in contrast to the nucleus accumbens, where if you knocked it out in this one cell type, they had a depressive behavior, but they retained the ability to respond to the antidepressant. So one of the things we're doing now is seeing if there's a possible connection between these two cell types. In other words, given antidepressant, it overcomes the damage you did from the knocking out in the nucleus accumbens, there has to be a circuitry that involves it, maybe third or fourth or fifth classes of neurons, but that's the way we're going with that. And so if you think of it, it's quite analogous to some other diseases. Parkinson's disease it represents the death of dopamine-producing cells. But you treat that disease right now by compensating for the missing dopamine and treating dopamine receptor cells. And this would be quite the same with these neurons in the nucleus accumbens are the reason for the depression, but you compensate by increasing the activity of these P11 cells in the cortex.
So we've had a conversation that spans 50 years of contributions from your lab. And I'd like to turn our attention to address a slightly different question, and that is more the social aspects of science. And that in addition to all of these legion contributions that your lab has made over the years, another contribution you've made is to train generations of additional scientists. And I was wondering if you wanted to comment about that aspect of looking back and thinking of the scientists who got their start with you.
Yeah, I'd be happy to do that. It's one of the things I'm happiest about in my career is that I've had the good fortune to have so many brilliant young people. Now, many people would say that people such as yourself and a couple of dozen others, what I did for you. I trained you to be great scientists.
And you did.
No, I don't think that's more credit than I deserve. I think all these brilliant young people realized how important what we were doing was, and they came to the lab because they were brilliant enough to realize how important it was. I've noticed over and over again that it's always the younger people who get the really important advances. For example, there's several times in my career, despite my experience along this line, where some young person has come along with something, a new way of looking at brain function. I'll say to myself, that can't be right. This doesn't feel right. It doesn't fit into my thinking about the brain. And they were right. And I think that's what happened with me. A lot of older people didn't believe in it, but the younger people did. And so they came and studied with me and learned our trade and went off and did their own brilliant stuff. But I didn't turn them in. I might have helped them be more effective scientists, but they had brilliant minds to begin with, like you.
Thank you. But one of the things, I still, after all these years of being in the lab, and I think it's been over 30 years that I've worked in your lab, will still go back to things that I learned from you. Not only about how to analyze scientific data, which is also true, but in particular, how to work with the younger scientists in my lab and get them to reach their own potential. You were particularly good at it.
Tell me what I did, because
it must be a critical ingredient that I have been trying consciously and purposely to identify, and I'm not sure that I've yet identified it.
Well, I think in my case it's a problem because first of all, there's a correlation between the talent of the people and how often they want to talk to me. You'd think it'd be the opposite. The neediest students and postdocs would come more often. They don't. The ones that are most talented would love to come and talk about their work with me. So that's one observation which I wouldn't have predicted to be the case. I think the problem is when you have a very talented person and they want to go down this path and you think it's better go down that path, should you let them go down the first path rather than the second path? And I tend to do that, and it's usually wrong because I've had so much experience that you develop an intuitive sense of what are the more productive avenues. But once in a while, they were right, and it's been led to very exciting stuff. So I don't think one can generalize. I don't know how you solved the problem. I haven't solved it.
No, I would agree, actually. I think that makes a lot of sense.
Yeah.
It's also made your lab very dynamic from the point of view, thinking back to what we were talking about earlier, and the resistance of the field of neuroscience to some of your initial discoveries. Your lab has not resisted new discoveries. Your lab has continued to embrace new discoveries, new technologies to a very impressive degree, and thereby remained at the cutting edge.
Yeah. Well, maybe I shouldn't have said, no, that can't be right. I should have said, I don't think that's right, but let's see whether it's relevant to what we're studying.
Yeah. Let's talk also about your early years in science and what got you started yourself. One of the things that I realized when I was your graduate student was how unusual it was for somebody to take your path, to go from industry and then having a spectacular career in academia. I've known many people who've done the opposite. And I was wondering if you could take us through that early part of your career. What first prompted you to go to industry, and then what led you to go back to academia?
All right. I went into industry because I was always excited, since I was a student, with the idea that one might take one's knowledge of basic science to develop drugs to help people. And it seemed a very exciting possible way to go. And then I was asked to become the head of this division of what at the time was Geigy. Then they merged and became Geigy, and then they merged it with Sandoz and became Novartis. And so I went.
Roughly when did you go there? Was that late 50s or early 60s?
1959. Yeah. I actually officially joined them in 1958. I'd spent 5 postdoctoral years studying in England, and I joined Geigy at 1958 and was there till 67. But the first year the laboratories weren't ready, so I went down and worked with Sidney Udenfriend at the NIH. And then I went into the industry, and I was in a pretty senior position for a young person, and was the head of one of the departments. There were 3 departments. There was a biochemistry department. They hardly used the term molecular biology then. There was a pharmacology department, a synthetic chemistry department, and then a medical department. And then there was somebody who was like primus inter pares amongst us. He kind of led the thing. But every time I had just about every time I had a new idea about how to develop some drug, you couldn't start a program unless the committee agreed. And virtually the committee never agreed. So some of those I did anyhow, and they looked very exciting, but I got so frustrated by the time that I left in 1967 that I just said, I think I have a better chance of contributing to drug development in academia than I did here at Geigy. So, in fairness, the situation is very different now. There are many more exciting opportunities now. In those days, the synthetic chemists ruled supreme. They were the gods. And all they wanted to do in those days was to make chemical modifications to somebody else's patented drug so that we could get a drug that would make more money. There was no biological thinking at all. And I didn't realize that they would be so obtuse. Now the situation is very different, and usually it's biologists in charge now. And in those days, when I was there, the biologists were the servants of the chemists, and now it's kind of turned around.
And then when you made the transition back to academia, you did so through a series of sabbaticals?
No, I had been, the last 3 years of my 5 years in England, I was studying at the National Institute for Medical Research. And there were 2 guys there named Murdoch Ritchie and Bill Douglas, who were both prominent pharmacologists. They had moved to Einstein about the same time that I'd moved to Geigy. I really loved England. It was a peak of McCarthyism, and it wasn't so pleasant. And I'm not politically active, but I think, and I didn't like to see the people I knew were being investigated by the McCarthy Committee. It was kind of, people forget how it was really unpleasant. And I enjoyed England, and I'm kind of a non-boastful type. And English tend to be non-boastful, so I felt more comfortable there than competing with the boasters here. So I really loved it there, but then after Sputnik, money started really going into the U.S. science, and it didn't in England. So, for example, I had to develop my own fluorometer to develop a very powerful new method for assaying what were called intermediate metabolites. So I decided I had to come back to the States for professional reasons. So I worked at Geigy for these 8 years, and then I decided to leave, and I took a job at a place which wasn't built yet on Staten Island called New York State Institute for Mental Retardation. I think they've changed the name. But it wasn't built yet, so I had a sabbatical year, basically. And I spent 6 months at Albert Einstein and 6 months at Vanderbilt. Albert Einstein, Murdoch Ritchie, and I, my old colleague from England, and I were working.
Was Murdoch the chair of pharmacology?
No, Al Gilman was the chairman of pharmacology. And it was really probably the best department in the country at the time. And Murdoch and I worked out the mechanism of action of local anesthetics. In fact, I was just subject to an interview about how we discovered them. We showed that the local anesthetics cross the plasma membrane of the axon in the neutralized form, and the acidic milieu inside the axon made them protonated, and so they couldn't get out, and they sat in the sodium channel and blocked it. It was just a fun thing. Anyhow, at about the time that I was leaving Geigy to go to the Staten Island Institute, Ritchie and Douglas were asked to be finalists for the chair of pharmacology at Yale, which Arnold Welsh had. He was a cancer chemotherapy guy. And they asked Ritchie what his conditions were. He said, if I will accept the chairmanship, I can bring Douglas and Greengard. And Douglas said, I'll accept the chairmanship, I can bring Ritchie and Greengard. So at this point, they offered me this. Actually, Ritchie won he's the one called, because when Douglas was asked what he would do, he was asked by the then dean, what would you do about this dead wood in the department? He said, I'll tell you what I would do with them. There's like 6 professors of chemotherapy. He said, I'd put them on a raft, I'd get in a motorboat, and I'd tow them out to the middle of Long Island Sound, and I'd cut the rope and come back to New Haven Harbor. And you always wonder why Ritchie got the chairmanship over him, but I know from the dean that was the reason.
It's funny.
So,
having had both of them teach me pharmacology, I could see Bill Douglas doing that.
Can't you see that? Yeah, that was Bill Douglas.
And then
yeah, that's how I ended up at Yale.
So that's how you ended up, and then you moved to
I almost went a year earlier.
So you moved to Yale in '68.
Yeah.
You almost moved a year earlier?
That was a point that was going to be in the psychiatry department. And there's some people strongly for me, but one powerful guy opposed to me. He said, no.
What does this have to do with psychiatry?
Yeah, basically.
Yeah.
He was a very well-known guy whose name I will not.
No, but you proved, but you proved yourself correct and them wrong.
Him wrong.
Him wrong. Let me ask you some even earlier questions in your training. You were born in New York City?
Yes.
What high school did you go to?
I went to 2 high schools. I went to first I went to Richmond Hill High School. And after 2 years, they had started Forest Hills High School. And then I transferred there.
So you grew up in Queens? Forest Hills, Queens?
Yes. Well, I spent the first 10 years of my life growing up in Brooklyn, then moved to Forest Hills.
Okay.
And they had very few high schools then. It was a long, long walk to go to Richmond Hill High School. It's not quite like Abe Lincoln, but it wasn't so good.
There was electricity then?
There was electricity. But then after the 2 years of Richmond Hill High School, they built the Forest Hills High School, so I went to that one. That's a school in which Raymond Simon and Garfinkel had this song, when I think back on all that crap I learned in high school, that was Forest Hills High School.
Oh, that's amazing. Wow.
Yeah.
And then you went from Forest Hills to Hamilton College.
Correct. Well, I spent 3 years in the Navy.
Oh, that's right. Yeah. So, what did you do while you were in the Navy?
Well, I volunteered to go into the Navy. I mean, unlike all the wars since, it was a pretty black and white issue there, and everybody hated Hitler, and so all the kids my age practically volunteered to go into the war. And in the Navy, I was sent to this electronic technician school after boot camp. They basically did a kind of filtering for performance ability. And then we went through 3 of these electronic schools that were more and more complex. They took the top, few from each school. And at the end of that, I was sent to MIT, where they're developing early warning radar system.
Mm-hmm.
The reason they were developing it is that they had these kamikaze planes that would fly just 20 feet above the surface of the water, and we couldn't detect them until they were like 20 miles out, and by then it was too late to get your airplanes up to shoot them down.
Mm-hmm.
They were torpedo bombers, so they didn't have much flexibility, just a thing carrying a big explosive, and our fighter planes could easily shoot them down. So somebody had the idea of putting a radar. It was very primitive in those days, the early days of television, a radar system in a plane, and then you could scan the entire thing. This is all then sent by what would be ultra primitive television down to the ship. And I was responsible for the electronics on the ship, as it turned out, and my buddy was responsible for the electronics in the plane.
It's amazing.
It was pretty exciting. I know I would have trouble with that responsibility. Now I have a whole naval squadron depending on you.
Yeah, but
and it was an 18-year-old kid. But
it illustrates.
At that age, nothing bothered you.
And it illustrates your quantitative nature and the proclivity you have toward physics and engineering. Did you study that at Hamilton?
I majored in physics and mathematics at Hamilton College.
Mm-hmm.
Yes.
And did you enjoy your college years?
Yeah, I enjoyed them very much. Well, I was catching up, so I only spent 2 years at Hamilton. There were a lot of older men and kids who took accelerated courses because we lost 3 years in the war.
Mm-hmm.
So I, went there when I was 20 and took summer both years, or took extra courses, and graduated when I was 22.
I see. I think I told you this, but a number of years ago when I was bringing my children on their college interviews, we went to Hamilton, and I was thrilled to see in their new science building a display devoted to you and your contributions as a famous alumnus of the college. I'm sure you've seen that.
Yes. I was there and I saw it, yeah.
Yeah. And then from Hamilton, you went to get a PhD, and I believe you started at Penn, but somehow made your way to Hopkins. Could you tell us how that happened?
Yes, well, at that time, I switched from my interest in physics and mathematics to biophysics, medical physics. And the reason for that was this is like 3 years after the dropping of the atomic bombs on Hiroshima and Nagasaki. And I thought, I don't want any talent I may have to be used for that sort of thing. Mm-hmm. And my roommate in college was the son of 2 physicians. And they both told me about this, and I was expressing my concern about going to that area, and they were telling me about this emerging field of biophysics. And so I learned about it, was quite excited about it. And what they were doing is studying the electrical properties of nerve cells, which is where we started the beginning of this thing. Everything was electricity. Mm-hmm. Old people working on the brain, they were electrophysiologists. There'd be biochemists using the brain for the research, but it was only because of a very rich source of enzymes. They were interested at all in what was in there. Mm-hmm. So anyhow.
So you went to Penn initially.
So I went to Penn, and I got into biophysics. There were 2 such departments in the country. One was doing radioisotope tracing at Berkeley, and the other was the biophysics department at Penn, which was chaired by a guy named Detlef Bronk, who then moved 1 semester after I got there to Johns Hopkins to be president. And he took with him one of the people, a guy named Keffer Hartline, who won the Nobel Prize for his work on vision. He became the chairman of the department. And then they moved a few of us with them from Penn to Hopkins. So I spent the last 4.5 years of my graduate studies at Hopkins.
And your PhD then was in biophysics?
Biophysics, yeah. I was studying the properties of nerve cells as they degenerate.
Mm-hmm. And then you mentioned earlier that you then spent a few years in England as a postdoc. With whom did you work there?
I worked with 3 people. One was a man named Henry McIlwain, who's best known for developing the McIlwain chopper. But he also...
This is a brain slicer where that was like a deli slicer, but to cut brains in thin slices.
And he made a couple of other significant contributions, mostly methodological. And then from there I went to University of Cambridge, or Cambridge University, where I worked with a guy named E.C. Slater. Right after I got there, he moved to Amsterdam, so I spent a good part of that second year abroad working in Amsterdam on oxidative phosphorylation, which is his specialty. I wanted to learn more biochemistry because my degree had been almost exclusively in electrophysiology. And then after that, I moved to the National Institute for Medical Research. The reason for that is I wanted a department that had equipment, facilities to do both biochemical and electrophysiological studies. There was no place.
It's very unusual in those days for the brain.
The only place there was a few pharmacology laboratories, which had some biochemical potency. And that was the head of that group was a man named Wilhelm Feldberg, who was a very, very important person in the history of pharmacology. He and Henry Dale were kind of the giants in neuropharmacology at that time in England.
So let's finish, if you would, just to give us a perspective of what you see moving forward, what you see in terms of how some of the many discoveries that your lab has made over the last several decades eventually influencing medicine. You alluded to some interest today, actually directly, work done directly by your lab in the area of Alzheimer's disease and depression. More broadly, many of us talk now about the difficulty that the field has had in introducing new medications for neurologic and psychiatric diseases.
For half a century they've been known as drugs.
Right. So I was wondering what your perspective is on that.
Well, it's clear, and you know this very well, Eric, the reason why the same old drugs keep appearing is because they figured out to some extent how these drugs work, and so they keep using the same targets to develop better drugs. And there's been substantial success, less toxicity, more rapid action, longer onset, longer duration of action, and so on, but basically nothing important conceptually. Now that we've learned all these things about signaling pathways, I plan to spend whatever number of years I have left working trying to do what's called today translation research to understand how our knowledge of these signaling pathways can contribute to understanding diseases and the drugs that treat those diseases.
Right, and you gave us some examples earlier in Alzheimer's disease and depression, but also more broadly, as you mentioned, in Parkinson's disease and schizophrenia.
Well, another example is Parkinson's disease. Nat Hines and I, with an extremely talented postdoctoral fellow in our laboratory, Miriam Heyman, in my laboratory, developed this trap technology where it's now possible to look at all of the proteins being expressed in every individual nerve cell type in the brain, the transcriptome, if you will. And what we're doing with it now is one example. In the case of Parkinson's, it's known that in Parkinson's disease, there were two types of dopamine-producing nerve cells. One group in the substantia nigra and another group in the ventral tegmental area, which are anatomically very close to each other. And we're now analyzing all the proteins expressed in the two types of nerve cells, and we're finding large numbers of differences between them. And why this is exciting and potentially of great therapeutic interest is we're hoping that we can find cells in the substantia nigra that are very abundant there, and they identify those and then put them into VTA cells in new mice and show that you can cause the death of those mice, those of the dopamine cells in those mice. And conversely, and much more practically important, take proteins which are very abundant in the VTA and make mice where you put them into the susceptible cells in the nigra and see if we can protect those cells from neurodegeneration. And that technique is an example of how these modern molecular biology techniques can be enormously helpful in understanding how the brain works.
It really transformed the field of neuroscience.